Good morning and welcome. I'm Al Koritz, Director of Applications and Support for Electron Microscopy Sciences. I will act as moderator for the series of webinars that we've been producing. I would like to ask you to take time to respond to our brief poll during the webinar or afterwards. Electron microscopy sciences and quorum technologies together have many decades of excellence in sample preparation. Today, we present the third in our series of webinars examining 3 distinct sample preparation workflows for electron microscopy. Today's tutorial, presented by Dr. Helena Bland, is about cryo-FIB-SEM techniques for lamella preparation and volume imaging. Dr. Bland joined Quorum Technologies as an application specialist in 2024. She completed her integrated master's degree in chemistry with biological and medicinal chemistry at the University of York. Following her degree, she worked as a research assistant at the University of Helsinki, specializing in carbohydrate synthesis. Helena completed her PhD in chemistry at the University of Sussex, where she synthesized leptin peptides and investigated their impact of the aggregation of amyloid beta and tau using TEM. In her role at Quorum Technologies, Helena delivers technical expertise and guidance to the company's global customer base, working closely with users to understand their specific workflow requirements and recommend tailored application-based solutions. Through her support, Helena helps customers optimize the performance of their systems, adapt to evolving scientific and technical needs, and achieve reliable, high-quality results in their work. Without further delay, I give you Dr. Bland. Helena, it's all yours.
Thank you so much, Al, for that lovely introduction. As Al just mentioned, this is our third in our 3-part series on electron microscopy sample preparation techniques. And today we're going to be talking about cryo-FIB-SEM techniques for lamella preparation and volume imaging. There we go. As Al just mentioned, EMS and Quorum have been working together for over 20 years, and we are specialists in electron microscopy sample preparation. This includes critical point drying for the dehydration of samples prior to SEM, glow discharge systems for the surface modification of TEM grids, sputter coaters and carbon evaporation systems for the coating of non-conductive samples prior to SEM, and finally, what we're going to be focusing on today, which is our cryo systems for the rapid freezing and preparation of samples in their near-native-like state for SEM. Just to give you an overview of what we're going to be covering in this webinar, we're going to start off just by defining what cryo-FIB-SEM is and how cryo is useful in FIB-SEM workflows. We're then going to move on to a more detailed overview of how our cryo-FIB-SEM processes work using our PP3010 cryo preparation system. Then I'm going to be talking a little bit about cryo-FIB-SEM lamella preparation and volume imaging, and then finally answering some frequently asked questions that we get at Quorum Technologies and EMS, and then opening the floor to questions from any of the audience. Just so we all start off on the same page, I'm just starting with what is cryo-EM. So cryo-EM is a technique that allows us to observe samples in their near-native-like state by rapid freezing at cryogenic temperatures. The idea is that we're able to preserve our sample while it's hydrated, so we're actually seeing more accurately what that sample looks like. You can see on the right of the screen, we have an image of a springtail antenna. And of course, if we were looking at a springtail in ambient SEM, we would have to first dehydrate that sample before we put it into our microscope. With cryo, what we can do is we can rapidly freeze that sample and keep it at cryo temperatures to allow us to see that sample in its hydrated state. There's a few benefits associated with cryo-electron microscopy. The first is that it eliminates any artifacts associated with chemical drying, critical point drying, and freeze drying. There's a really nice example of this on the screen. I'm just going to get my laser, where we can see with our freeze drying, we are seeing these artifacts where the structure has collapsed. Whereas when we're looking at our cryo-prepared sample, we're able to maintain that structural integrity of this stomata. Cryo also allows us to image delicate samples, including that may be typically removed by other methods of dehydration. So for example, with critical point drying, we use solvents, and this may strip off certain delicate features from our samples. Cryo is also really great for time-lapse imaging. So what we can do is, if we were, say, observing a manufacturing process, we can freeze frame at each point of that particular process, and then we can look at those on our SEM. So it's a really good way of sort of capturing something as it is happening. Finally, it's useful for reduction of beam damage on beam-sensitive samples, for example, lithium-ion batteries. So then what is FIB-SEM? When we have FIB-SEM, we're combining two beam columns, where we've got our electron beam column for our imaging, and then our focused ion beam, which can be used to remove material from our bulk sample. So we're using a stream of ions which can cut, mill, or sputter away thin layers of our material in our SEM. This beam is typically either gallium or plasma, but you can also have helium and neon ion beams as well. So what's great about FIB-SEM is we can do a couple of different extra things that we wouldn't be able to do without our ion beam. We can do micro machining, where we're using our focused ion beam to pattern surfaces on a nanometer scale. We can cross-section samples, so if there's something that we want to reveal below the surface of our sample, we can mill away at that sample to reveal a section further down in the sample. This cross-sectioning also combines with the next point, which is that we can do 3D reconstruction. So by milling a sample and then taking an image, and then milling that sample again, taking another image, we can use those combination of images to do volume imaging, where we combine those images to create a 3D representation of that structure. Finally, we can do lamella preparation for TEM imaging. So we can use our FIB beam to produce thin sections that can be removed and then imaged in our high magnification and higher resolution TEM microscopes. So what happens when we combine the two techniques together? So obviously, the obvious benefit of combining cryo with FIB-SEM is that it now allows us to do FIB processes with hydrated structures. So for example, if we were wanting to look at a hydrogel, say we wanted to do some volume imaging, if we were to put our hydrogel in ambient conditions, we would have to first dehydrate that hydrogel. And ultimately, a hydrogel that isn't hydrated is no longer really a hydrogel. So what we can do is we combine having our cryo stage at approximately -150 degrees or below, and we can combine this so that we can remove our layers of our hydrogel, we can image, and then we can produce a 3D structure of that sample. Cryo also helps to improve stability during high-dose ion electron exposure. It can also help to stabilize components that otherwise might outgas or deform. And finally, it allows us to do lamella preparation for cryo-TEM. So we can have a full process where we have a frozen sample, and we can go from cryo-FIB-SEM all the way into cryo-TEM. So what would that process actually look like? So our PP3010 cryo preparation system is what we call our full cryo preparation system. So the idea is it can take you all the way from freezing in our slush nitrogen freezer over here to preparation and finally imaging in our SEM. The whole system is cooled using gas lines that are supplied by a pressurized dewar. There are 3 gas lines that cool 4 components of the system. Everything is controlled using automated software on our prep deck. And as I mentioned, there are 4 different components that are being actively cooled. In the preparation chamber, we have a stage, and then in our SEM, we also have a stage that looks like this. And these are both being cooled to a minimum of -140 degrees. But typically with, if you're doing particularly delicate work, you might be looking at using -150, -160 degrees on your stage. We then have an anti-contaminator in our prep stage and an anti-contaminator in our SEM, which act as a cold trap to remove any moisture from the chamber. So these would typically be set at least 20 degrees below our stage temperatures. And these can be cooled all the way down to minus 190 degrees. The gas enters through a cryo heat exchanger, which contains liquid nitrogen. And then this is used to cool those gas lines, which cool these 4 components. There are some additional features that we can supply with our PP3010 preparation system that are particularly useful with cryo-FIB workflows. The first is our rotate stage. So our rotate stage allows 360-degree rotation, which makes it easier for locating regions of interest and positioning our sample to the correct position for FIB milling. And in addition to this, we have an auto refill system. So our standard system has a runtime of 24 hours. But when we're doing particularly long processes like cryo-FIB-SEM to TEM workflows, it might be that we actually want to keep our sample longer and guarantee it cold for a longer period of time. This is because if we're doing, for example, fibbing overnight, we might do some automated fibbing. We might then want to continue the process the next day, or for example, we might prepare our lamella and then we've got the lamella, but we're not actually ready to go into our TEM because maybe someone else has booked it, or maybe it's got towards the end of the day and you're not mentally prepared to do your TEM. So what you can do is you can keep your sample cold in the system overnight, and this helps to reduce any contamination that's associated with putting samples just in liquid nitrogen. And this system can run for a minimum of 48 hours. The way the system works is we have a sensor on our cryo heat exchanger. So we'll automatically top up our nitrogen that's cooling our lines. And then we also have a much larger dewar, so 120-liter dewar, to supply our gas lines. And I say minimum 48 hours. Whenever I talk about the auto refill system, I like to quote our world record holder for the longest cryo runtime. So this graph here was produced by one of our collaborators, Chris Parmenter at the University of Nottingham. And he has ran a cryo system using this auto refill for a total of 106 hours continuous runtime. So I always say if there's anyone who is feeling competitive in the audience, then get yourself an auto refill system. And 106 hours is currently the time to beat. So what does the whole workflow look like of going from cryo, freezing the sample, into our FIB-SEM, doing our lamella preparation, and then moving into our TEM? So I'm just going to get rid of this laser pointer, and there's a video that's hiding in the screen. Oh, I'm just going to turn the sound off for that. So this is our shuttle that we use for our processes in our cryo systems. Shuttles can be specially designed for different sample types, and this particular shuttle is designed to hold a TEM cartridge and a planchette. So this is where our cryo-TEM cartridge sits, and then our planchette will sit in this area at the front. We are assuming in this particular example in the video that the sample has been high pressure frozen prior to transferring into the system. So our system does have a slush nitrogen freezer, but it's also compatible with high pressure freezing and with plunge ethane freezing. So if you prefer to freeze your samples not in slush nitrogen, you can do that and then transfer over to our PP3010 system. So the first thing we'll do is we'll load our shuttle into our slush liquid nitrogen. So we'll get that shuttle cold for putting our high pressure frozen sample onto our shuttle. Going inside that TEM slush pot, you can see we've got our slushing station, which has our slush liquid nitrogen. And then we have our preloaded HPF planchette, which is then transferred over using tweezers under the slush nitrogen into our planchette holder. This is then secured using our specialized clamp so that the sample is put securely in place. And once we have the sample loaded, we can then move it over to our preparation chamber. Everything sort of transferred in the system is done using our transfer pot, which is this that's coming on the top now. The transfer pot is used to retrieve that shuttle, which now has our planchette inside it. And the shuttle is pulled up into a compartment in the top of the transfer pot. This can then be sealed under vacuum during the transfer process so that the sample is not exposed to air. I'll just pause and you can see that this is the separate compartment that our shuttle's been retracted into. It's sealed, and then we can transfer this over to our preparation chamber. The transfer pot sits on the preparation chamber. We then pump down the airlock to make sure there's a good enough vacuum, and then we can transfer our shuttle into our PP3010 preparation chamber. In this chamber, there's a couple of preparation steps we can do. We have fracture blades. There's one on the front and one on the top. If this was just a regular cryo-SEM sample, you might just use it for fracturing. But in this case, we're using our knives in order to open our TEM cartridge holder. And if we have two planchettes on top of each other, we might fracture the top planchette off. We can then do our sputter coating. So if we have a non-conductive sample, we'll sputter coat that before it goes into the SEM. And then once our preparation processes are complete, we can push the sample into our FIB-SEM. Inside the FIB-SEM, we then have our actively cooled rotate stage, which will be at least minus 150 degrees, and we can start to do our milling of our sample. So we'll identify our region of interest and then typically use a gas injection system to coat with platinum. And then we can cut our lamella using our FIB. What that typically looks like is we will produce two separate trenches on either side to isolate a section that's mechanically stable. And then we'll start to do thinning towards that section to make it between 100 and 300 nanometers. Once we have our section, we can then lift it up with a micromanipulator probe and place it onto our TEM grid cartridge. So now we have our lamella loaded from our bulk sample onto the cartridge. And then we can transfer that back into our preparation stage. So that's simply just pulling it back from the SEM into our preparation stage. And then at this point, what we can do is we can close that holder so that we secure that TEM grid nice and safely inside the, with the lid closed. And then we can pull it back out with the transfer pot. From this point, you can then move back to the slush nitrogen and transfer onto your TEM holder. So that's what the process looks like. And here's some images that are produced from the University of Nottingham, where we've been doing this process. So you can see that we've got our lamella attached to our micromanipulator needle here. We've placed it on the side of our TEM grid. And then our lift-out grid, sorry. And then we have transferred over and we have a TEM image of lamella. In addition to lamella prep, cryo-FIB-SEM can be used for volume imaging, which is where we take an image and then we mill the sample, take another image, mill the sample again, and keep repeating until we have a sequence of images that allow us to reconstruct a 3D model of that particular sample. And there's a really interesting example of where this has been done in the literature. So this paper written by Andrei Shabayev et al. Is really interesting and shows the use of the PP3010 in a cryo-FIB milling cryo-SEM process. So what they've done here is they have nanoparticle-free polymer composites that incorporate ionic liquids. Obviously, if we wanted to look at ionic liquids, we wouldn't be able to do this without the added function of having cryo. So this is a really nice example where cryo is essential to the process. And what they've managed to do is they've managed to identify where the ionic liquid typically occupies in these polymer composites, and that's typically in the amorphous region. And they've also managed to show that this excess ionic liquid can increase conductivity. So this is a really nice method of being able to demonstrate a strategy for tailoring polymers and ionic liquid composites. So I said that I would answer some frequently asked questions that we get a lot at Quorum Technologies and Electron Microscopy Sciences. And the first one is that how long does the process typically take? And obviously, it will vary depending on the instruments you have. So different SEMs have different automated processes for lamella lift out and thinning, etc. But typically, so this is from Chris Parmenter, where if we sort of do the worst case scenario, we have 8 hours and 15 minutes to do the full process, which really emphasizes the importance of having a system that can run for a very long time, because this obviously would be continuous working and we can't always continuously work through. So this having a auto refill system that allows us to have our system running at cryo temperatures for a very long time means that we don't have to rush through our particular processes. The next question is how to avoid ice contamination in our cryo-FIB-SEM workflows. So I've got an example of sort of large ice contamination on the left and some small ice contamination on the right. And this is mainly associated with storing in liquid nitrogen. So if at some point during your transfer, you're having to store your sample in liquid nitrogen, then you are potentially introducing ice crystal contamination to that sample. So a huge benefit of being able to keep your sample in the cryo preparation chamber is you won't see this sort of ice buildup on the sample. And then also it is important to note that if you are doing FIB processes, to an extent you can remove some of this ice contamination with the FIB beam itself, but obviously the less contamination you build up, the better the sample is going to be. So thank you so much for listening to our talk today. This was the final one in this particular series. So to keep up to date with any future webinars, please visit the EMS website on the link below. And if you have missed any of the previous parts of this webinar series, we have been uploading those to YouTube, so you can watch those videos on there. Thank you so much, everyone, for listening. Just open up the floor to any questions.
Helena, thanks for the fantastic presentation. Can you easily process samples that were frozen by other means, like a grid in a vitrobot, or is it just high pressure frozen samples and such?
Yeah, so this example was just showing the high pressure frozen sample transfer, so with that particular holder. But you can do your freezing. There's sort of three main ways. Well, there's more than three ways to freeze the sample, but three main ways that we talk about at Quorum, which is, so we have our slush nitrogen freezing, which is what is built into our PP3010. And we say this is good for about 90% of samples. If you have a particularly thick sample and you need to have good freezing throughout the bulk of the sample, then we'd recommend you either do liquid ethane freezing or you do high pressure freezing. This is because those two methods are able to freeze at a faster rate compared to our slush nitrogen. So if you were doing plunge freezing, so for example with a vitrobot, we have different shuttles that you can transfer to get it into our system. And then if you're just doing slush nitrogen, then you're already in our system, so there's no need to do the transfer. But yeah, you can freeze in a number of different ways, and it's all compatible.
Great, thanks. A question from the participants. How do you deal with ice contamination on samples, since FIB-SEM chamber vacuum is normally no better than 10 to the minus 5 Pascal? Do you sublimate the sample before performing cryo-FIB-SEM?
Yeah, so we do. I didn't actually mention it, which was silly. I should have done. So our cryo preparation chamber has a feature for sublimation, and it's automated, and you can set the temperature between -80 and -100 degrees. And what that's doing is that's heating up the stage in order to remove any water vapor that might be on the surface of that sample which could cause ice contamination in the SEM. So yes, I would always recommend that you sublime your samples before they go into the SEM, just in case there is any small layers of water on the top of that sample that you might want to remove before you're putting it into the SEM.
Another question from the same individual. We might have touched on this, but not really answered his particular question completely. Can you contrast the differences in plunge freezing and liquid ethane and liquid slush? Or not just liquid nitrogen without the slush? We know that just liquid nitrogen is really bad for freezing.
So liquid nitrogen without the slush, there's this effect called the Leidenfrost effect, which is that when you put something into liquid nitrogen, you get this insulating gas layer on top of that sample. So it's much slower freezing because of this gas layer in liquid nitrogen compared to slush nitrogen. Slush nitrogen is also at a much colder temperature, so it has a faster freezing rate. So liquid nitrogen would be the worst thing to freeze with. Then slush liquid nitrogen would be, it's colder, so minus 210 degrees. You'll get a faster freezing rate because you have less of this Leidenfrost effect. Then one step up from that is liquid ethane freezing. So that has a faster freezing rate. And then high pressure freezing is the sort of fastest way to freeze. But it is kind of sample dependent. So a lot of samples you don't necessarily need like high pressure freezing. For sort of regular cryo-SEM work, most of the work I do in the lab downstairs, I will just use slush nitrogen freezing, and it works for a lot of samples. So it's very sample dependent.
I just might add that there's a very good paper out there, although it's sort of old like me, by Dr. Joseph Costello at the University of North Carolina, Chapel Hill, who discusses and contrasts and shows the difference in plunge freezing in different cryogens and such. It's, an old paper, but it discusses and demonstrates the different processes and which is advantageous and which is disadvantageous. Next question. What are some drawbacks using cryo-FIB-SEM?
So some drawbacks. Well, with cryo, you are typically working with very sensitive samples. And with cryo, actually, you might be more prone to artifacts like curtaining on your FIB-SEM. This is because basically ice, if you have any ice at all, it can impact, it sort of causes non-uniformity of milling. So you're having to be more gentle with samples, definitely, with cryo samples, but that's just because of the nature of the samples, not necessarily the fact that it's cryo. So I guess the drawback is, that you might be prone to like more curtaining on your images.
Thank you. A little follow-up. How does the lamella adhere to the TEM grid after picking up the section using the probe?
So that's related to, so before we do, there's sort of in FIB systems, you'll have a gas injection system where you'll platinum coat, and it's the platinum basically that welds it to the probe.
And the last question in the queue here, how does sublimation process affect samples with high water content?
So the higher the water content, typically the longer you need to sublime samples for, but it does really depend what you're wanting to see from your sample. So we have I don't think it's online actually, but we have a very interesting video at Quorum that shows just sort of the sublimating process. You can see sort of the water being removed from the sample as it happens and revealing those sort of internal structures. So the sublimation process is, yeah, essentially removing that water vapor. So the more sublimation you do, the more internal structures you'll see. But if you do, you can essentially over sublime something. So if you sublime something for too long, it can cause, for example, salts to crash out of solution, and you'll get something called eutectic ridges. So you can start introducing artifacts into your images that way as well.
I just might want to add again, since I've been doing cryo for a very long time, there's a number of elegant papers describing sublimation rates in vacuum by Dr. Heinz Gross at the ETH in Zurich. He did a study. Also, Martin Mueller of the same institution. They did a number of papers exploring freeze fracture and characterizing different processes like sublimation in vacuum, which is really no different what we're doing in cryo-SEM. It's just easier now looking at the samples in cryo-SEM. Before, you used to have to make a replica of your cells after you fractured them and sublimate before you coated them, and then float the replicas off for TEM use. The resolution was quite nice, but we're definitely approaching or exceeding that resolution level now in field emission microscopes using the PP3010. But those are two individuals that sort of really pioneered a lot of the freezing work, as well as categorizing sublimation rates at different vacuum levels in these systems. You can figure it out down to angstroms per second of water molecules leaving. They did a very elegant paper where they used deuterated water and isotopes of water with the hydrogen being in isotopes. This way they could use a mass spectrometer and differentiate between sample water and background water, so they get very accurate sublimation rates for that particular study. Is there any other questions? Wait. Yes, there's more. How can you be sure that freezing is fast enough to ensure sample vitrification and there's no crystal formation during the freezing process?
So again, it's sample dependent. But you'll the more cryo you do, the more you'll get used to seeing what is an ice artifact and what is actually part of your sample. So I guess you know that you've frozen it fast enough if you're not seeing artifacts. Yeah, I guess I don't know if you've got anything to add to that, Al.
Yeah, this is simply a well, okay. For the biological folks, this is a lifelong problem. Having achieving great vitrification for processing your samples, but for the most part, the high pressure freezing done correctly has really eliminated this. And as far as on the quorum prep system, as long as you're aware of your depth of freezing and you limit what you're trying to freeze, you can achieve vitrification down at least 5 microns or so or better, depending on the sample. Using cryoprotectants is really not a good idea, because that changes sublimation rates, as well as puts some artifact. Dr. John Rash of the University of Colorado in Fort Collins used to do a little cryoprotecting by using pure ethanol and to remove some of the water from the sample, sort of a little dehydration step. but it really didn't alter the results. And he was doing freeze fracture of cells. So, and that would be the most gentle cryoprotection you could use that would work with this particular workflow, I think. Next question. Have you experienced platinum implantation into biological or any other soft material when depositing a protective layer by cryo-FIB for lift out for TEM? If so, do you have a workaround for this?
So yes, kind of. So well, one way that you can sort of minimize is by sort of prior to if you sputter coat the sample in the chamber beforehand, you are kind of putting down a physical layer that can act as like a barrier to prevent the platinum ions from implanting directly onto the specimen. So having that sputter coating on top of it allows you to do, like prevent that slightly by having that physical barrier. Otherwise, I don't think I personally have any suggestions.
Yeah, I'm really surprised that, well, okay, not totally surprised, but normally you would see ion implantation, one of the older coating techniques is obviously, ion sputtering and such. But the interaction between the FIB beam and the sample is really difficult to mitigate. You need that high energy to remove your material that you're interested in removing. Having platinum interact with the beam and end up attached to your sample is an unfortunate byproduct, but I don't really have a way around that either. That would be maybe a question for the FIB-SEM manufacturers. They may have more data on beam sample interaction than I have. Let's see. All right, a little bit of a shift. How about freezing? Is freezing beneficial for cryo-FIB-SEM on batteries or material samples compared to biological samples?
Yes. So, I mean, we have mostly talked about, although I did talk about polymer composites, biological samples today. But yes, cryo-FIB-SEM is very useful in particularly battery research. One of the reasons for that is because you have liquid electrolytes in batteries, so like lithium, and to keep those sort of in the phase that they would be in, you want to use cryo techniques. You can also, lithium batteries are highly sensitive to exposure to they're very beam sensitive, so having cryo is also beneficial for that as well. So yeah, batteries is actually a really good example of something that works, like is better with cryo-FIB-SEM than without it.
Yeah, if you're talking about, sample and beam interaction mitigation, certainly cryo is the way to do that. Simply, you're imparting quite a bit of energy into a very, very small area, and having that area cooled to mitigate some of the heat damage is very beneficial. I think this is the last question, unless somebody slips one in here. Would you be able to check lamella quality before transferring to the TEM for observation?
Yeah, I mean, you can use the SEM, and also if you have a STEM detector to look at that thin layer. Obviously, you won't know what it looks like in the TEM because you still have a transfer process to do before you get there. But yeah, to an extent, you can check it a little bit in the SEM.
Yeah, I agree with Helena. That's possible. I was thinking STEM detector also if the microscope is so equipped. However, um, checking it before the transfer, is a good idea simply because a lot of samples are lost in transferring to the TEM. At least you know where uh, the the pain point is in your process. You have a good sample, after fibbing, and you're in the transfer mode, and somewhere along the line, by the time it gets to the TEM, it's not looking so good. At least you're able to isolate and try to troubleshoot the poor sample at the TEM side. What we do is not easy, and takes a lot of skill and effort to get great results. And that's what this whole series of webinars has been about: getting great results, optimizing your workflow, understanding the different workflows and how it can benefit your sample. I enjoyed this series of webinars. I've learned a lot along the way. I thank Helena for brilliant presentations along the way. And with that, if there's no other questions, we'll bring this series to a close. We will probably have some more content of this type later on this year. Thank you, everybody, for attending, and feel free to reach out to us if you have any further questions, or we can assist you with your workflow. Have a great day.
Yeah, thank you, everyone.